Good afternoon, everybody. Welcome to the August webinar for H3 Bionet, which is organized by H3 Bionet Seminar Series Task Force, or other coordinators that organize monthly webinars throughout the year. For this month, the theme is Viral Genomics, and the title of the talk is Viral-ngs, Cloud Compute for H3Africa's West African Viral Genomics Centers. The seminar talk will be provided by Dr. Daniel Park, who's a senior group leader for Viral Computational Genomics at the Broad Institute. This talk will be for 40 to 50 minutes, and it will be followed by a 10 to 15 minutes time for question and answer as well as discussion. I'm kindly going to ask, if you're not speaking, please mute your mic so as to not disturb the speaker, because for some of you, there's some noises in the background. So kindly mute your mic if you're not speaking. And just to make sure that we have some order for this webinar, I'm kindly going to ask you that if you have a question, either type it in the chat box if it's burning, and Dr. Park will answer it, or you can raise your hand. There's a top little thing looking like a human, an icon on the top there that you can use to raise your hand, and obviously it will get the attention of either myself or Dr. Park. So just to make sure that we have some ground rules and we do not disturb the speaker. The abstract is a bit long, so I'm not going to read or even read a bit of that because I'm looking at time as well. We only have about an hour. I'll send an ad. If you want to read the abstract, you can go to the ad again. But just a few lines from Dr. Park's CV. Dr. Daniel Park is a senior group leader for Viral Computational Genomics at the Broad Institute. He works along Prof. Paddy Sabeti and Dr. Bronwyn McInnes, where he co-leads viral genomic sequencing and analysis efforts at the Broad, including genomic epidemiological analysis of viral outbreaks such as Ebola, Zika, Lassa, and unidentified viral fevers. Since 2013, Dr. Park has led computational analysis and methods development in the group and has directed the implementation of the computational pipelines for use at the Broad and by the consortial partners in West Africa. Dr. Daniel Park earned his PhD in organismic and evolutionary biology from Harvard University in 2013, where he developed and utilized computational tools to identify adaptations in PFAS from malaria parasite that allow it to evade modern drugs. He joined the Broad Institute as a software engineer in the year 2006, supporting microbial sequencing efforts. He earned his master's and bachelor's degrees from the Massachusetts Institute of Technology, or rather MIT, in 2002 and 2001, respectively, both in electrical engineering and computer science. Without further ado, Dr. Daniel Park, thank you so much for giving us your time and effort to give this talk. Go ahead and give a presentation.
Okay, thank you. And thank you just for all the organizers for inviting me to give this seminar. I'm looking forward to kind of connecting with the H3 Bionet community to talk about particularly what we've done in the H3 Africa context with their consortium in West Africa. This is my first time using Adobe Connect, so if I'm not seeing a hand raised, hopefully it'll pop up or something, but if not, just let me know if there's something that needs to be addressed. So just to give a little bit of context, the purpose of what we're doing with viral genomics in this consortium in West Africa is manifold, but it's really kind of comes down to the question of what can the genomes of viral pathogens tell us to improve the treatment and the kind of control of viral disease. And there's lots of different ways. I'm not going to really kind of go into all these types of things, ranging from just knowing what's there and what's circulating to more translational things and even more epidemiological applications. Some of these, and again, I won't quite go into, are kind of more obvious today in 2020, even though the patterns of analysis are quite similar to what we've done with previous outbreaks. What I'm showing you is a little bit of an older picture of the types of analyses you can do with viral genomes back from the Ebola outbreak in West Africa. And the basic idea is if you have pathogen genomes or viral genomes in this case from an epidemic, you know, these genomes, there are standard ways to go from that to phylogeny, which basically tells you ancestry and relatedness of all the different individual pathogens. And if you're taking this from a whole bunch of human patients, inferring ancestry and relatedness gives you, inference into transmission and epidemiology. So this is something that you've probably seen pictures like this, not just during the West African Ebola outbreak, but subsequent Ebola outbreaks in Central Africa during Zika, and of course a lot these days during coronavirus. A lot of the approaches are quite similar. This is something that our consortium has really kind of pursued for the past decade or more, with a focus on hemorrhagic fevers such as Lassa virus, and the areas in West Africa where that's endemic. It required a kind of build up a lot of specialized laboratory and computational abilities to deal with things that are kind of special to these types of viruses, and ultimately resulted about 7 years ago in the launch of an H3Africa funded project centered, led by Professor Christian Happy, who I'm sure you all know quite well, at Redeemers University, but including partners at Cheikh Anta Diop in Senegal and the Kenema Government Hospital in Sierra Leone, as well as U.S. partners such as myself. And the purpose here was really to bring genomics, which back in 2013 was kind of a new idea, to research centers in West Africa that were studying viral disease and febrile illness. And as part of this, there was an effort to bring stand up lab capacity and data generation capacity within these 3 research centers. This kicked off right around the time of the West African Ebola outbreak, which underscored the importance of what we were doing, but also in some cases added some logistical challenges. So, for example, in Sierra Leone, things kind of got kicked off. Had to wait a little bit until the worst of it was over before some of the equipment could really go into place. But this is kind of how long these kind of pathogen research institutions have had genomic sequencing capabilities on the ground. But part of this, and me personally, I was brought into this project at its inception back in 2013 to address the question of, it's one thing to put machines and lab capacity and the ability to generate data in West Africa, on the African continent, but very closely coupled to that is how then are we going to enable these researchers to analyze their own data and to pursue their own research questions without having to lean on outside institutions such as the Broad Institute, for example, to do a lot of this analysis for them. And so being part of a group that had developed various informatic methods to address, tricky genomes like, highly diverse viruses, probably a familiar story to many folks in bioinformatics, this originally was a relatively informal pile of scripts at one point, about a decade ago, and was also very hard to share with any institution, really, let alone with our African partners, just because a lot of these tools had been highly tuned to our particular compute environment. And so I had kind of started on the project of trying to figure out how do we make these informatic tools more portable and usable almost as immediately as these machines started to come online with our partners. And so part of what happened so this happened in 2014, and partly motivated by the West African Ebola outbreak. We had partners at a company called DNA Nexus that worked with us to try to figure out how to extend this analytic ability to our H3African partners in West Africa. DNA Nexus, if you're not familiar with them, is probably the oldest genomic cloud compute company out there. They don't normally deal in pathogen genomics. They usually deal with large kind of cancer projects and things like that. But they had an interest in allowing their platform to be used in kind of a broader, um, I guess for them, almost a more inspirational way. And so they worked with us to help wrap a lot and port a lot of our tools to their particular platform, which initially was in a more kind of corporate proprietary way. So it was kind of specific to their particular platform. But I'm going to be talking more in this presentation about how we've moved into a more portable representation of that. But it basically, and I'll go more into what the tools and workflows themselves actually do scientifically, but these are the basic types of, whether it's, just quality, QC, and more mechanical things to metagenomics and assembly and phylogenetics. These are the types of things that you might want to do if you are sequencing viruses and generating data. They had a pretty nice and mature web interface for launching off jobs, looking at the results, and looking at various different kind of outputs of that. That's something that they had established for some time. It's not unique to them, but this is what it looked like. And it essentially, it made it so that a lot of the more standard analyses that we do computationally were more of a web kind of point and click interface, right? And it extended the ability to pursue these analyses to a broader set of folks than just, say, the traditional bioinformaticians. And so what was interesting was at the outset of this project, we didn't initially think we were going to be going for a cloud-first approach, but by experimenting in this space, I think we learned a lot about the advantages that it afforded us. Now, ultimately, a lot of our tools are first and foremost open source tools. You can get them online at GitHub. You can download them and get the either Conda packages or Docker containers and run them on the command line on your own hardware. And this is something that anyone can do, but actually we find that most people don't actually run it that way. That the cloud wrappers, the nice platform that not just makes it easy to execute, but also organizes a lot of the data for you and gives you access to a wider range of hardware to run some more kind of special types of analyses. It really opened up access to the compute to a much broader range of users. And I mean that in two ways. It's not just that it opened up the ability to do this with our partners that actually had kind of less infrastructural resources. So if you didn't have a standing compute cluster in your lab, you could get up and running anyway because this was all on the cloud. You didn't really need a lot of setup. So that's part of it. It facilitated access to a wider range of institutions without having to put a lot of hardware prerequisites on the ground. But sorry, was there a question out there or no? But the other thing that we found was it also provided access to a wider range of just users of different types of training and background. And so for us, this was more apparent even within our own lab at the Broad Institute, where, 10 years ago, I think we were a bit more of a traditional shop where we had kind of a partition between our laboratory people and our computational staff.
Sorry to disturb. There's a bit of an echo. Someone is in the background speaking. Sorry to disturb. Sorry, can we please all mute our mics because that's feedback. So can you all mute your mics? Otherwise, I'm gonna have to mute everybody's mic myself because it's disabled the speaker. Okay, sorry about that.
Not a problem. Okay. Let me know if you need to do a quick mute. But yes, so what I was saying earlier was in our own group, we had people who generated data essentially in the laboratory and then people who analyzed it, and there was the computational person and say, hey, can you run this quick analysis for me? And there was a bit of a back and forth. What we found, though, is that once we started providing these tools in a web platform, in a cloud compute manner, even within our own group, it really increased the ability for our laboratory researchers, the people who know the science but maybe not the command line, right, to be able to answer a lot of their own questions. And there was a lot less of this kind of partitioning, actually, as a result. There were a lot more folks who were fluent in both the analysis and the laboratory protocols as a result. Anyway, this proceeded to actually, as part of the consortial efforts, as part of our H3 Africa project in West Africa, it facilitated a lot of kind of nice little projects and viral analytic projects by folks in our consortium. Up until this point, a lot of the genomic analyses were really led by folks over at Harvard and the Broad Institute in the US. But from kind of starting probably around, this is probably the first paper in 2016. So Nigeria, if you recall, had their own little Ebola outbreak during the West African epidemic, but it was much smaller than a lot of those other countries. But it made for a nice small sample set that you could study on a MySeq and do some basic analyses on. And so this is kind of the beginning of a number of different small vignettes and stories and papers that have come out of our consortial partners, really led by analyses that they were able to do themselves on these platforms. From there was other, there was a yellow fever outbreak, there was a Lassa outbreak, there was, these are kind of Nigerian examples, but there were further kind of studies that happened in Sierra Leone and then Senegal. Some of these were very kind of scientific in nature, some of them were more kind of public health oriented. Of course, these days it enables a lot of the work that is happening around coronavirus, for that has come out of the centers here that have been sequencing them. So for us, it's been it's kind of proved itself as a very enabling tool. So it kind of shifted us to realize that actually, when it comes to computational pipelines, prioritizing cloud as a primary way of providing access to compute was working really well for us. But of course, there's a lot of questions and concerns that usually come up, and certainly that we had, that made me skeptical 7 years ago about whether this whole thing was going to work. And of course, the first thing that a lot of folks ask is, well, what about things like power and internet? If you are doing compute on the cloud, don't you need internet? And I think part of the example of what we've seen there is just looking at kind of our own experience. This is the Professor Happy's lab at Redeemers. It's just like the router kind of network logs for his lab for some random week in the fall a couple of years ago that I just took a snapshot of because we had access to that data. And this kind of shows a relatively typical network pattern that may be familiar, where there are times of day where the internet uplinks that are provided by the university are really quite nice and quite fast. In fact, this is kind of a sliding average, so it smooths out some of the peaks, but I know for a fact that there are times in the middle of the night at Redeemers where their connection is actually quite, much faster than what I get in my home in Boston. But then there are other times where it slows to quite a crawl. And then there are other times, and you might be able to see this in some of the gaps in the lines here, there are other times where it cuts out completely, either because the network just goes out or the power goes out. And the question is, what do you do with that? If you use a relatively naive tool to try to upload a sequencing flow cell to the cloud, it will not survive a network connection like this. But if you actually look at how much bandwidth is available to you over the course of, averaged over the course of a whole week, what you realize is that it's actually more than enough to handle the average output of a sequencing lab, even the one at Redeemers. And so really, this is a problem that just needs to be solved with software, which is something that DNA Nexus helped us with. Probably about 5 years ago is when they implemented a solution that essentially just stubbornly uploads data and assumes that the connection may go up or down and takes advantage of the times where it is actually in good shape. And it's telling that it's often able to get out flow cells to DNA Nexus just fine at times when even at times when the lab is having a hard time holding a Zoom call, that particular day. So it's not as much of an issue as we'd originally thought. The hardest part, if you do most of your genomic compute on the cloud, is just getting data to the cloud. But after that point, the advantage of doing it on the cloud is that you only really need your internet connection for control of your execution, so to launch jobs and maybe to examine some results, which are relatively low bandwidth. And if you need to, you can do it on a mobile device. I've certainly done that myself. It doesn't require moving lots of data around, and it doesn't require stable connection, because once you've kicked off 1,000 assembly jobs or something, your power can go down, and it doesn't really matter because it's running just fine in a data center somewhere else that someone else is maintaining. The other question that people have asked a lot is, what about the costs of cloud compute? Because the costing model is different and seemingly less predictable. I think there's a lot of kind of concern about that. What we've actually found is that in a way, there are aspects that are more predictable. It's a little bit, it requires a little bit of just running things regularly and seeing what you get. But for a lot of the more standard, predictable kind of analyses that people will do, if the code for the workflow is mature and things like that, we find that they often cost a similar price range every time you do it. And so you can kind of predict computational cost as proportional to the quantity of data that you're doing, which actually means if you are a African sequencing center, it should be proportional to the amount of data you're generating. Now, these are prices, example prices for some assemblies, which is not the whole workflow, but it's just part of, it's an important part of it, on DNA Nexus and the Broad's Terra Cloud Platform. And they're listed in US dollars. And they're a little bit different, actually, partly because, well, I mean, it's the same code, but they're running on, DNA Nexus is running on Amazon and Terra's running on Google, and those costs are a bit different, and some of the efficiencies are a little bit different. But you can see that the order of magnitude here is just far, far, far less than the cost of generating the data in the lab, to the extent that it becomes almost trivial, really. So for us, I think we found it to be not nearly as expensive as actually running a sequencing lab. On the flip side, actually, we found that it saved us, in a way, a lot of money, partly a lot of startup cost. I think I mentioned before the advantage of cloud compute is that you don't really have to set up too much local infrastructure. There's not as much kind of prerequisites to kind of get started there. But a lot of times when it comes to running things, whether you're running an HPC or just a large single shared compute box or something like that, there's a lot of hidden costs that usually aren't taken into the equation. It's not just the cores and the RAM and the hard disk, but especially the cooling and especially the power. I mean, if you're running a genomic sequencing center, you still need to have the power redundancies and the inverters and things like that. But if you are running a compute cluster, you need far more. A large compute machine consumes far more power than an Illumina sequencer does, right? And then on top of that, there's the personnel cost as well for administrating an HPC or something like that is is kind of the specialized role, and these are roles and costs that we've managed to avoid so far at our different sites in West Africa. Another concern that often comes up is around the question of who's able to see my data. And part of this really just comes down to learning each platform. It does kind of depend a bit on the cloud solution that you end up pursuing, but there's a bit of fear, especially if as data leaves a physical location, whether that means that there's kind of less control over the access to it. Our own experience with this, I think that, so I just want to say this is a very important concern that really has to be thought about carefully, but I think has been thought about carefully by certain vendors and platforms that actually have to meet certain regulatory requirements. So if you are a DNA Nexus, or if you are a Terra, or if you are one of these types of things that have had to go through regulatory compliance processes, then what we found is that actually the controls, the privacy controls around this are probably actually better than what you would get if you kept your data locally, and I'll go into that in just a bit. Our own experience with DNA Nexus has been a bit telling, and with Terra as well, in that we often find that there are other users from other labs who are trying out our viral pipelines on their own data, and sometimes request help from their respective help desk, the DNA Nexus help desk or the Terra help desk, because they're experiencing some failure or something like that. And if this is a platform that is run properly, this is something where the support desk, the admins, none of these people should be able to have access to any of that data unless someone is explicitly sharing with them. So for example, if you want the help desk to help you with a particular failed run, a lot of times they can't just go looking at what it looks like unless you somehow give them permissions on it. The one exception that sometimes people forget about who does have access to the data, other than you, the people who upload it and the people you share it with, are the people who pay for the data storage, and that's kind of a natural extension of if someone is paying for the storage of data, then I guess it's kind of seen as they are allowed to see what that data is. But I think I mentioned before, what we found is that these costs tend to be negligible enough that it, for us, does make sense to pass the cost ownership onwards, so that the full control of the data can be kind of kept within the country and within the continent where the data was generated. On the flip side, I mentioned this before, I actually think there are actually some far less kind of controls on the data sharing and the security of it when you keep data in a more traditional context where you compute in your own cluster and things like that. I mean, any typical Unix setup basically affords the administrators of that setup to be able to see all of your data. Now, they are within your own institution, so hopefully, those are folks that you trust. But more importantly than that, in a traditional setup where data sharing literally equals data copying, what that means is that there's not really an ability to revoke the act of sharing data. And so whoever you share data with can make their own decisions about who they share data with, and you can't take that back. And so I think there's actually a way in which having it live in a controlled environment actually gives you a bit more control over how far it can get away from you, in a sense. I'll try to go through this part a little bit quickly because I don't want to go actually too much into the guts of almost the science of what it does. But I mentioned this whole viral analysis suite. And I'll just kind of briefly say there's a bunch of bits I think I mentioned before that have to do with kind of this is more reflexive data processing, things that don't require too much human interaction, so just demultiplexing, base calling, QC, stuff like that. We do, since we study non-human, we study microbes out of human patients, host depletion informatically as part of our standard process, and kind of metagenomic classification. I think I mentioned earlier we have a bit of a focus on febrile illness, and since there are many causes of febrile illness, metagenomics is an important part of the research here. And then there's also, the assembly of genomes, which kind of gets you to the phylogenetics and things like that. These are things that require a little bit of user interaction because whereas the first part, when you run a sequence flow cell on a sequencer overnight, a lot of times you can get this automated set of reports the next morning. Here you got to kind of tell it, what am I assembling? What are the knobs and the parameters I'm going to use for these types of analyses? And so this is the part that we train scientists on and how to perform. I won't go too much into this, but it's not just the kind of raw outputs, but there's visualizations that are kind of associated with this. I think I already had this in the slide earlier. Visualizations also around the phylogenetics as well. This is some of these pieces are a little bit newer than others. We've been adding features year by year as different scientific needs come up. Different ways of pulling and pushing data from data repositories and things like that. So that's the type of tool sets that exist in there. So I wanted to just end a little bit with a little bit of a run through of what's under the hood and how we write the code. These are some of the general principles that we try to keep in mind as we build out this system. I can't say that we check all these boxes yet, but I can say that as the years go by, we are checking more of them. But there's a whole kind of separate line of thinking around how best to kind of create these modular interoperating bits and pieces of an analysis platform. There are many organizations that have spent a lot of time thinking about how to define APIs and standards around this, and I think many of you are probably familiar with a lot of these types of things. But it's basically, how do you put together an ecosystem where a lot of these different players are probably going to be different entities. The people who write the tools aren't the people who run the tools or the people who generate the data, or maybe these are the same people, but this is someone, that type of thing. So I think this is what we've been trying to move towards, and you can kind of start to see some of these principles as I kind of run through this. When we write our code, there's kind of two layers that we write it at. One is we write these like wrapper scripts around certain units of work where if for those of you who are deep into the bioinformatics, you know that like everybody wants to use this one aligner, but that one aligner has a bunch of annoying little edge cases that it just fails on. I mean, most short read aligners and snip callers out there seem to fail when you give them, for example, if you give them 0 reads, right? Or just things like that, right? So there's always the kind of annoying fun of file format conversions between that tool's output format and some other tool's input format. So we try to wrap a lot of this into things, and these are the things I mentioned before where you can run it on the command line if you want, and if you don't want the whole wrappings of the web platform. But the second layer, and kind of one of the important layers on top of that, is how do you describe the workflow itself? And I know that this community is relatively familiar with, there are several ways and languages that have kind of emerged as some of the best practice ways to describe an informatic pipeline in a portable way. Several years ago, we shifted a lot of our stuff to one of the languages called WDL, or the Workflow Description Language. Happens to be created by the Broad Institute, but actually that's not the reason that we ended up adopting it. The reason we ended up adopting it is actually because our existing partner, Nexus, was very excited about this particular language and had been shifting a lot of their stuff in that direction. And so several years ago, we shifted a lot of our kind of proprietary language pipelines from Nexus into this workflow description language, and that became the way that we maintained our pipelines and delivered them to that platform for the past several years. And I'm showing you a bit of code simply to say if once you're in a language that you like, you realize actually it's actually quite nice when a workflow like this, and I'm showing on the left a pictorial kind of flowchart of the de novo assembly pipeline, is actually kind of concisely describable with all of this code here on the right. This is the whole pipeline, the same pipeline that's kind of described there on the left. So that's what we've converted a lot of our stuff to since then. More recently, if you don't know about Docstore, this is something I've been evangelizing a bit more in certain informatics communities. But what's nice is GA4GH has put a lot of thought into how to define the buckets of different components of a large platform. There's the parts that execute, the compute. There's the parts that hold the storage, and then there's the places where you maybe hold and distribute a lot of the tools and the workflows. And so Docstore is kind of the premier implementation of the GA4GH Tool Repository Service API, or TRS. And it's actually really quite nice. This isn't just like a bunch of diagrams on a screen. I'll show you like how it actually kind of works for us in kind of a more practical sense. But the idea is, like, if you've got your workflows in CWL or WDL or Nextflow, and actually they just recently added Galaxy support as well, this is a place where you can kind of upload them, describe them, and distribute them widely, and a lot of other execution environments can hook into it via a backend API and just allow users to launch your workflow in their platform. And so in our case, this year we recently converted all of our Travis continuous integration kind of setup where, every time we make changes to our WDLs in GitHub, this is now building and pushing those changes to Docstore. And what that means is you can go to look at any one of our pipelines on the Docstore page and just click with a button and launch it on a number of different popular kind of cloud platforms for genomic compute. Or you can download the workflow and run it locally, which is pretty normal. But this level of integration is actually surprisingly mature, and it's for us it's our primary way of integrating with the Broad's Terra platform. The other thing that's nice is this system is an independent entity, right? And they give the ability for organizations, you can register a new organization, to go in there and curate collections of tools. And this is one of my ambitions for, I know that Alan came and spoke about a month ago or so on the Phage organization, but this is one of my ambitions for one of the things that Phage can do, which is to, sometimes maintaining a repository of the best practices of tools for some like, is it, what are the best tools out there for, drug resistance calling and TB or something like that, right? Sometimes you want a neutral entity to curate that collection versus the people who actually, write all the different tools. And so this kind of provides a mechanism for that. You can see all of our different tools here at this link down on the bottom. But this is our particular set of collections. Anyone else can kind of curate their own set. Let's see. I don't want to spend too much more time here, so I'll try to wrap up quick. But what's nice is I feel like once you start working a lot in in trying to distribute pipelines in a cross-platform way, you kind of learn a few principles about how to write that code in a way that tends to work. And for us in the Viral group, you know, I mentioned before, for the past few years, we initially were pretty DNA Nexus based. Recently, we've become much more Terra based, but we know for a fact that this also works just fine on local environments through other different types of executors of the pipeline. In theory, it should work on a number of different, you know, on your HPC or on Azure or something like that, but it hasn't been tested there yet. But the lift to get from here to there is not as far as I would have thought it would have been. We are in a time at the moment where this type of portability is more accessible than it was a couple of years ago, I would say. More recently, we've shifted a lot of our stuff. I'm not going to really kind of go into this too much into the Terra platform. It offers a few advantages over DNA Nexus, but a lot of it is conceptually quite familiar and similar. It's a web-based platform where you can house your data and execute compute, and it runs on top of a commercial cloud. But it comes with a kind of an in-house support system. It comes also with a better ability to do notebook-based analyses and aggregate kind of visualizations across a bunch of different runs. There's our own team has been learning it a lot more during the course of this past year, and there's definitely a number of different upsides to that. So there wasn't a lot of changes we had to make to our code to make it work here, but it's been a nice shift, and it's kind of the direction that our group is moving. So yeah, it also kind of affords more ability to kind of self-document alongside the code and data, and there's just a lot more kind of instructional walkthroughs attached to our different workflows as we distribute them. I just want to wrap up to say that this is, as I mentioned, this has been a kind of long-running project over the past decade, and one of the important parts of this whole process has been the iterative kind of back and forth with our primary users, with our H3Africa partners in West Africa, in Nigeria and Senegal and Sierra Leone. Part of what we've done in that project, part of what my group has done, is not just kind of create these tools, but to run various workshops and training programs and just to support the analyses that they've been doing over these past several years. And each of those times, it kind of results in a bit of feedback of, oh, this could be better, or if only we had this tool, or if only this existing workflow had one more report or output with it, or things like that. And so there's just a lot of different lessons learned over the past 5 years or so of doing this, where we've found either because a certain site has asked for it, or because certain scientific projects have motivated it, we've just added either new features or different types of tweaks along the way. So yeah, that's all I have here. It's been a broad effort, and obviously a very big part of this has really just been the collaborative work in our H3Africa projects team in Nigeria and Sierra Leone and Senegal. I know a number of folks from those groups are on the call right now, and various partners we've had on the Boston side over the past several years. So I know there's a bit of bouncing on the thing, so maybe what I'll do is I'll stop sharing for now and see if we have questions. Let me see here.
Thank you so much, Dr. Park. Now it's question and discussion time. We have about 15 minutes. Nikki has a question. Go ahead, Nikki. Hi, Nikki.
Hi. Thanks, Danny. That was great. I was surprised to see that that there wasn't a major problem with the internet and using the cloud in Nigeria. Can you tell me what kind of science data sets were you transferring to the cloud in the first place? Obviously, viral genomes will be quite small, but have you tried sort of bigger human genomes or something, or what kind of data sizes were you working
Yeah, let me just quickly, let's see if I can go back to, I have some numbers on there. You're correct that part of what we get away with is the fact that in studying pathogen genomes, these are smaller. So when it comes to viral sequencing, a lot of times these labs will run maybe a dozen or 2 dozen different samples on a single MiSeq flow cell. So, you know, is obviously much smaller than a human genome. But we kind of calculated if they had actually run a MiSeq every single day at full clip, the amount of continuous upload bandwidth that would require is maybe about 0.5 megabit, which actually means, you know, if you have a 1 megabit connection on a good day, it could be down 50% of the time, and you could still be running your sequencer 24/7. So it does kind of depend on the type, the size of the data there. Now, initially, all of our different sites, these are kind of one sequencer shops at first. At this point, you know, Christian's lab has 2 MiSeqs, an iSeq, a couple nanopores, I think, and they have a new HiSeq. Now, we haven't turned the HiSeq 4000. I think it's turned. We haven't tried this upload piece yet. I think we got to put a bigger hard drive on that laptop. I'm not too worried in that there are. This is if it comes down to it, you can buy your way out of, you know, the problem. But so far, this is not something that has really caused a problem. Like I said before, other things start to break down with the internet, such as video calls, well before the sequencer breaks down.
Thank you for that. Peter, go ahead.
Hi. Sorry, I was just going to audio on this side. So yeah, I kind of had a follow-up question to that. So you mentioned that you did some Plasmodium Amplicon sequencing. Was that just for the resistance gene region? Because I know that Plasmodium samples tend to be, in my measuring, about 1 gig per genome. I get what you're saying about running multiple samples on one flow cell, but I'm wondering if this approach is pretty much limited to viral genomics.
I see Ala's on the call, and I don't know if she can maybe comment on the loci. It was a multi-locus Amplicon approach. I believe, and as you know, Peter, we have discussions around doing more malaria genomics this way with Gates in these different with these different partners. To my knowledge, I believe we don't yet have ambitions to do whole genome sequencing locally, though 1 gig isn't too bad. It depends actually more on how often you're doing it. And certainly in Dakar, their internet is not as bad as it is in like Sierra Leone, but I think our ambitions at the moment are more of a multi-locus kind of approach, both for drug resistance and also for just kind of diversity monitoring. Ala, are you? Do you want to speak to that at all? If not, that's okay.
Yeah. So yeah, sorry, I have a weak connection today. Can you hear me?
Yes, we can hear you.
Okay. So we were able with some students to run this Amplicon sequencing using the CDC SOP for drug resistance marker. And now we have this complete SOP ready to run many sample on one flow cell. So like we can run 20 sample for using just one flow cell. So this approach is really, I don't know how to say in English, is ready to be used, and we can share it with you if you like.
One of the things that we thank you.
One of the things
one of
that
the things that we found.
One of the things that we found.
Thanks, Ala. So one of the things that we found from the Harvard side that has done a lot of malaria genomics is that actually increasingly, depending on the question you're asking, it isn't always necessary to do full genomes. And I think we've increasingly been favoring getting a good set of loci, targeted loci, in an Amplicon panel that both hits resistance markers and allows people to surveil for kind of changes in identity by descent, because that is a good marker for the effectiveness of different kind of eradication effectiveness.
Sorry, Danny.
So.
Just to follow up, I mean the 1 gig wasn't a whole genome. It was, I think, 100 loci on that genome, because of the same problem of filtering out host DNA. People in general, as you know, don't really do a lot of whole genome on Plasmodium because you get all the host with it, and
right.
You have to get ridiculous depth, and yeah. But anyway, sorry, it just was 100.
Oh, okay. Thanks for the clarification. I think the way that I tend to think about it isn't so much how big is the genome, but how big is the flow cell or the unit of data generation, and how often are you going to run that? So if you know your sequencers that you have in your lab, what would be the bandwidth of that if you max them out all the time? And for a lot of these labs, actually, it turns out to be okay.
Okay. We still have about 5 minutes left. Are there any more questions or comments? If you want to type, you can type in the chat box, or again, raise your hand. There's an icon on top that you can use to press to get attention. Peter, do you have another question?
Yes, sorry. Just a quick question. Have you considered, Galaxy as a similar platform to what you're doing, or do you think that's a similar approach, or would you say that's different to what you're attempting?
Well, I think you're kind of the expert on that one. I personally, from what I understand of it, and Galaxy was something that I'd considered probably back in 2014 or so as well. It's conceptually, in my mind, very similar. I mean, it's doing a lot of the same types of things. It's a bit more. You still have to solve your own infrastructure, like how are you going to run that? But in terms of the and I think because of that similarity is part of the reason why a tool repository like Docstore has started to adopt that as well. So. Yeah, I consider that in the same similar family of solutions in my mind.
Thank you so much for that. Peter says thank you as well. Are there any more questions before we wrap up? We still have a bit of time, so if you have last 2 maximum questions or comments, and then we can close. I'll mute myself just to receive that.
Feel free to reach out to me separately if you do have questions. I think I had, I think my contact info might be there. Certainly my Twitter handle is as well.
Yes, it is. So I'll share the slides. I'll share your the recording, the link to the YouTube recording as well as your Twitter handle and your email as well, so people can email you or tweet you if they want more information. Peter is saying something in the chat box. By the way, Phage, I'm trying to let me try to read that.
I can speak a little bit to that, and I didn't plug this as hard as I could have. Partly because I knew that Alan was here a few weeks ago, but yes, the Phage Infrastructure Working Group is specifically thinking about a lot of these types of questions. Peter and I are deeply, part of what I was presenting today was just how our group had historically solved this. And I think this is a good vignette and a good model that people can follow. And I think as part of the phage infrastructure working group, we are interested in similar types of stories as well of different types of portable deployments that have worked well for people, particularly in Africa.
Thank you for that. One more question, and I'm gonna close. Is there any burning question or comment? And we can get Danny to answer that, or alternatively, as I said, you can separately send them an email, and we'll gladly engage with you. Okay, it's almost time. It's like 2 minutes to 4 p.m. In South Africa. So, Danny, thank you so much for taking time out and for using your enthusiasm to give this presentation. We're all looking forward to it. Do you have any comments before we finally say goodbye to everybody?
Not on my end. Thank you, everyone, for coming out.
Okay, sure. No, thank you so much to Professor Dr. Danny and to all the attendants for this webinar. I'm going to share the recording with you by tomorrow, and I'll see you next month for a different webinar with a different speaker. Enjoy the rest of your day, everybody. Thank you.
Thank you, everyone.